An international research team has generated the first truly complete sequence of a human Y chromosome, the final human chromosome, to be fully sequenced. The new sequence, which fills in gaps across more than 50% of the Y chromosome's length, uncovers important genomic features with implications for fertility, such as factors in sperm production. The study, led by the Telomere-to-Telomere Consortium, a team of researchers funded by the National Human Genome Research Institute, NHGRI, under U.S. Department of Health and Human Services. The study has been recently published in the journal Nature.
The Y chromosome, along with the X chromosome, is often discussed for its role in sexual development. While these chromosomes play a central role, the factors involved in human sexual development are spread across the genome and very complex, giving rise to the array of human sex characteristics found among male, female, and intersex individuals. Additionally, recent work demonstrates that genes on the Y chromosome contribute to other aspects of human biology, such as cancer risk and severity. When researchers completed the first human genome sequence 20 years ago, gaps were left in the sequences of all 24 chromosomes. However, unlike the small gaps sprinkled across the rest of the genome sequence, gaps that the Telomere-to-Telomere Consortium filled in last year, over half of the Y chromosome sequence remained a mystery.
All chromosomes have some repetitive regions, but the Y chromosome is unusually repetitive, making its sequence particularly difficult to complete. Assembling sequencing data is like trying to read a long book cut into strips. If all the lines in the book are unique, it's easier to determine the order the lines go in. However, if the same sentence is repeated thousands or millions of times, the original order of the strips is far less clear. While all human chromosomes contain repeats, about 30 million letters of the Y chromosome are repetitive sequences. It's as if the same few sentences were repeated for half the length of the book. To tackle the most repetitive pieces of the human genome, the Telomere-to-Telomere Consortium applied new DNA sequencing technologies and sequence assembly methods, as well as knowledge gained from generating the first gapless sequences for the other 23 human chromosomes.
The complete Y chromosome sequence also reveals important features of medically relevant regions. One such section of the Y chromosome is called the azoospermia factor region, a stretch of DNA containing several genes known to be involved in sperm production. With the newly completed sequence, the researchers studied the structure of a set of inverted repeats or palindromes in the azoospermia factor region. Deletions in the azoospermia factor region are known to disrupt sperm production, and thus these palindromes could influence fertility. With a complete Y chromosome sequence, researchers can now more precisely analyze these deletions and their effects on sperm production.
Other regions with potential medical relevance contain genes that repeat. Most genes in the human genome have 2 copies, one inherited from each parent. However, some genes have many copies that repeat along a stretch of DNA, sometimes referred to as a gene array.